Hi, my name is Amane Takahashi. I'm a fourth-year student at Shizuoka University and a member of the DGTalAquaLab. In this video, I will present my undergraduate research in the form of a poster presentation. My research focuses on an invasive copepod in lake and the development of an individual-based model to study that species. More precisely, I'm working on a species that invaded Lake Taal, which is located on the island of Luzon in the northern Philippines and a caldera lake of the Taal volcano. This lake has experienced water temperature rising due to global warming and eutrophication due to intense aquaculture. And my research subject, Actodioptomus dorsalis, a neotropical freshwater copepod, massively invaded Lake Taal and many other Philippines lakes in the early 2000s. This species prefers food-abundant environments, so the eutrophic condition of Lake Taal, caused by aquaculture, is favorable for the settlement of this invasive copepod. Previous studies have shown that the production of this species is affected by temperature and food concentration. For example, as temperature increases, the duration in each stage decreases, and as the food concentration increases, the hatching success increases. These data are from laboratory experiments and can provide information about the effects, but only at the individual level. So, the purpose of my research is to create a model that can simulate the population dynamics of Actodioptomus dorsalis in Lake Taal, taking into consideration the effect of food concentration and water temperature on this species to understand what factors contribute to the settlement of this invasive species. Here, I present the first step in the model development, the implementation of the effect of food concentration on reproduction. Concerning the model I created, the model is an individual-based model. It was created using the Moby-Dick platform. The model simulates the reproductive cycle of egg-bearing copepods. The main parameters that the model considers are the following four: clutch size, the number of eggs in a particular clutch produced by an ovigerous female; embryonic development time, the time from egg spawning to hatching; hatching success, the percentage of successfully hatched nupli from each clutch; and latency time, the time from hatching to the next spawning. How each of these parameters is affected by food concentration is expressed by equations implemented into the model. Let's have a look at how these effects were parameterized. The data used for fitting the equations came from the studies of Tordeslas et al. In 2016 and 2018, and Elmore's study in 1982. We used the Bell-Headek equation for representing the effect of food concentration on LT and EDT, and the Hape equation for hatching success and clutch size. We implemented these equations into the model, and we verified the proper implementation by running the model with conditions similar to the experimental data. And here are the results of that model check. The conditions of the model were set to the same as the experiment conducted by Tordeslas et al. 2018, 30 degrees for different food concentrations, 5 females, and 15 days. The gray boxes are the experimental results, and the white boxes are the output of the model. The results showed a relatively good agreement between the model and experimental results, which confirmed proper implementation. Next, we increased the number of females to 1,000 and test the effect of food concentration on the total production of such a cohort. Let's have a look at the result of these simulations. The top graphs show the total number of eggs on the left and nupli on the right produced by the 1,000 females cohort throughout their life. At the lowest concentration, the span between one clutch and the next, called the interclutch duration, was long, and the females only reproduce once. But as food concentration increased, the interclutch duration shortened. Females start making eggs much earlier and produce many clutches throughout their lifespan. The number of nopli behaved similarly to the egg production, but that was lower than the number of eggs. This difference between egg and nopli production indicates that the model properly considers the effect of hatching success. And it shows the importance of food concentration on the production of the next generation for Actodioptomys dorsalis. The bottom graphs show the accumulated number of eggs on the left and the nopli on the right over the simulated period. The total production of both eggs and the nopli became more regular for food concentrations above 32,000 cells per milliliter. This results in more linear cumulative productions, as can be seen in the bottom graphs. In other words, 32,000 cells per milliliter appears to be the threshold for stable egg production of Actodioptomys dorsalis. If we look at the total production at the end of the simulation in relation to food concentration, we can see that the cumulated production of both egg and nopli increase as food concentration increases and tend towards the maximum. Half of the maximum production is reached for a food concentration of 27,000 cells per milliliter for eggs and 125 cells per milliliter for nopli. In conclusion, we already implemented the effect of food concentration into the model, and the current version of the model can already help us estimating the amount of food required for the species to have a continuous production. Next, we are planning to add the effect of temperature in this reproductive cycle. After that, we will create the rest of the life cycle, which describes the development from nopli to adults. That's all for my presentation. If you are interested in this lab, please have a look at the website. You will find many information including the contact of the PI, Dr. Duragail. And for more videos, don't forget to subscribe. Cheers!